Joanna.
Good morning.
We are here in this interview to evaluate your competences related to your evidences, which are ethical argumentation, explanation and functioning of chemical and biological systems, and the demonstration of the functioning of chemical and biological systems. So please share with us your work.
Good morning. My name is Joanna Marin Segura, and today I will present this proposal that is the proteomic evaluation of glucosinolate-enriched arabidopsis extracts on insulin resistance in a high-fat diet mouse model. In the previous evidence, the CYP79F1 gene was selected for overexpression in the Arabidopsis thaliana to enhance the biosynthesis of methionine-derived aliphatic glucosinolates. And while these compounds have evidence that can be converted into bioactive isothionates such as sulforaphane. And while type 2 diabetes affects more than 500 million people worldwide and is characterized by the insulin resistance, chronic inflammation, oxidative stress, mitochondrial dysfunction. And why is this important? This is important because several studies suggest that sulforaphane can improve these alterations. The hypothesis is that glucosinolate-derived isothionates obtained from the overexpressed plant can improve insulin resistance and by modulating the proteins involved in the insulin signaling, energy metabolism, oxidative stress, and inflammation. And the objective is to evaluate the antidiabetic effects of glucosinolate-enriched arabidopsis extracts using physiological and proteomic approaches. This is the general workflow. First, we have the harvesting of both the overexpressed and the wild type of Arabidopsis thaliana. Then we have the extraction that will be explained later, the administration to the mice, the physiological evaluation, tissue collection, and finally the proteomic analysis. For the extraction and characterization of the plant extracts, both the leaves from the wild type and the overexpressed arabidopsis plants will be harvested, freeze-dried, and subject to a methanol water extraction. And why? Because we need to do an standardization. Instead of feeding the whole plant to the mouse, they will receive these standardized extracts. And why is this important? Because we can have an accurate dosage control with an improved reproducibility, and it can be a better comparison with published studies. The administration will be via oral gavage, and why use oral gavage is because that way we can ensure that the mouse gets the whole dose. There will be used a high-fat diet-induced insulin resistance in a male C57BL6J mice. And why use this model is because it mimics the human insulin resistance. It's widely validated in diabetes research and is suitable for nutraceutical intervention studies. I propose the use of 5 groups: a control group with a standard diet, a high-fat diet group with nothing else, a half-fat diet group that will be given the wild-type extract, a high-fat diet group that will be given the overexpressed extract to see if the overexpression did something different from the wild type, and finally a high-fat diet group that will be given metformin to work as a positive control. And for each group there will be 8 animals. Why use 8 is because in previous studies evaluating this insulin resistance, they use about from 6 to 10 animals. 8 animals allow for a good statistical analysis, but still gives a rational number of animals. The workflow is from the week 0, the acclimatization, from week 1 to 12, high-fat diet induction and the confirmation of insulin resistance. From week 13 to 16, the treatment phase, where there will physiological measurements such as body weight, fasting glucose, fasting insulin, glucose tolerance test, insulin tolerance test, and lipid profile. And finally, in week 16, the tissue collection. For the proteomic strategic, we have three main steps. First, we have the protein extraction and quantification. The tissue from the liver will be homogenized using buffer and a protein quantification using the BCA assay. Then we have a trypsin digestion that cuts in smaller peptides, suitable for liquid chromatography and mass spectrometry. And finally, the liquid chromatography that separates peptides by chemical characteristics and mass spectrometry to determine the mass to charge ratio and to identify these peptides. And I propose the use of a Orbitrap Q exactive because it gives high resolution results. The target proteins are the ones involved in these 5 biological process. For insulin signaling, we have this, but the most important one is the GLUT4. For energy metabolism, we have the AMPK. For oxidative stress, the NRF2. Inflammation, the most important is the NFKB. And for mitochondrial function, these 3 are, the complexes are the most important. For the bioinformatic analysis, these are the software that will be used, and MaxQuant for the protein identification and quantification, Perseus that we'll use for the statistical analysis and to find the differential proteins, String and Keck for protein-protein interactions, and the pathway enrichment analysis. Finally, we have the ethical considerations that involve all of these. The 3 R's principle that talks about replacement, reduction, refinement, but also we have national and international regulations. From national, we have the NOM 062/1999 that talks about the conditions laboratory animals need to be. Also, we have the General Health Law and the Health Research Regulations. And from international guidelines, we have some like ARRIVE, Guide for the Care and Use of Laboratory Animals. And taking these regulations into account, we have the measures to keep the animals well by the use of minimum number of animals, in this case 8, daily health monitoring, control temperature and humidity, environmental enrichment, human euthanasia, and an ethics committee approval prior to study initiation. And well, these are my references. Thank you very much.
Yes. How are you gonna euthanize the mice? Which is the most human way?
While I was researching, I found that the most human way is by using a gas in gas cameras to do it. And in this case, it doesn't intervene with the study, so that can be used to do it in a human way.
And how are you gonna standardize the doses that you're gonna give?
I'm gonna go back. The plants will be freeze dry, and with the methanol water extraction, they all will have the same concentration. So that way, it can be compared. And yes, it can be compared. There is also the characterization of the plant, so we can know like what does it have, but with the same concentration, we can standardize what we are giving the mice.
Well, my last question. Do you think that it is necessary that you have like previous experiments in order to know the doses that you need to put in the mice? To the mice?
Yes, there will be necessary to have some previous steps before.
Like which?
Well, for starters, the characterization, because we need to know like approximately how much the extracts have to know how much of the plant we are giving the mouse, and well, yeah, most.
But why?
Why? Because why? Because that way, well, because we're working in animals, we can do like a lot of repetitions because it takes a lot of animals. So we need to have like a previous something to know if we are working like in the right ranges, to know to not like do experiments in without como en falso.
Okay. For this, how can you don't do these unnecessary experiments in mice? What can you do previous to these experiments in mice to know which dose will function to avoid inflammation or other things related to diabetes?
Well, there could be like experiments previously in cell lines to prove the extracts and see if they're responsive. First do it in cells, and if they have a good response, then pass it to animals, and that way we can have like a idea of how much of the dose we need to give the mice.
Okay, do you know any cell line or cell type that you will be good for this type of experiments?
I remember. I think they're called HEP2, that it's about the liver, and it's a chronic inflammation model, and that could be a good model to evaluate the extracts.
Okay, thank you, Joanna. I have no further questions. Okay, that will be all.
Thank you.